So we're kind of winding down a little bit. In this unit, we're going to study the urinary system, and it's the only system we'll study for this unit. But we're going to follow it with chapter 21, which is going to pull together a lot of the things you've learned about body fluids throughout A&P. And then our last unit will be, we'll start after this one, of course, and that will be on the reproductive systems, and then we'll be done. So, on the urinary system, this one doesn't have that many organs in it, but one of the organs is very, very detailed, and that's the one we're going to look at today, the kidneys. And we won't even finish with the kidneys today. But the purpose, the big purpose of your urinary system is to remove chemical waste. So when it says waste, that's of course not talking about digestive waste. It's cellular waste products or metabolic waste products from chemical activities in your cells. CO2 is a metabolic waste product. That's removed by your lungs, but pretty much everything else can be removed at your kidneys. Okay, and then so it's moving all of that from the blood, so it basically filters your blood of the things you don't need anymore, except for CO2. And then the resulting fluid, of course, is urine that you're very familiar with. And then from the kidneys, that urine moves into a ureter. So you have two kidneys and a ureter leading from each kidney. That's not a ureter. It's a ureter, okay? And then that transports the urine to the urinary bladder. There's just one of those, of course. And then from there, the urine moves through the last organ, the urethra, that transports it to the outside. So basically, the ureters, urinary bladder, and urethra are simply to transport or store the urine. And so the kidneys would be the big organs, because as far as function is concerned, because they filter the blood of the things you don't need. Okay, so here you can see where everything's located. Do you notice the left kidney sits up a little bit higher than the right kidney? And just for fun, let's think about lab. When we name blood vessels, what is that blood vessel's name? Good, the abdominal aorta. What is this blood vessel's name? The inferior vena cava. Very good, very good. Okay, and so here's an artery that feeds the kidneys. That's the renal artery. And so that blood then is filtered through the kidneys. And then what the cleaned up blood that by now is deoxygenated moves into a renal vein to go into the inferior vena cava. So that's a little renal circuit. A drop of blood does that about 60 times every day. So this is a great filtering organ. And then for fun, what is this blood vessel? That's not labeled here, but you learned it in lab. That was the first one that came off anteriorly from the aorta. After the aorta penetrated the diaphragm, it starts with a C. It was a little bitty short one that then branched into the upper abdominal organs. It's called the celiac artery. And then this would be the superior mesenteric, and this one is the inferior mesenteric artery. So that's just for lab review. Okay, students struggle with those a little bit. All right, but getting back to the urinary system, here's a ureter, and then of course there's the urinary bladder and the urethra. All right, so I like this picture. This is showing you a transverse section through the upper abdominal area. The A picture is showing you that. And if you'll notice what I'm outlining in green, this is the parietal peritoneum. So that's the lining of the abdominal cavity. Do you notice that the kidneys are located behind that? Okay, and embedded in some adipose tissue, some fat. All right, so kidneys are not out there in the abdominal cavity with the liver and the stomach and the intestines. They're located behind the cavity wall. And so that's a very protected area. Here on this second picture, you can see a lengthwise section of that. So there's the parietal peritoneum. Okay. Right. Okay, and then so there you see a highlighted view of the kidneys. And again, this would be the celiac, superior mesenteric, and inferior mesenteric arteries. You can see that. Okay, so you can see they're in the upper part of the abdominal cavity. This word right there, retroperitoneal, that means behind the parietal peritoneum. Okay? Behind the parietal peritoneum, retroperitoneal. Okay? All right, and the left one's up a little higher than the right one. The kidney is one of the several bean-shaped organs in the body, and so that indented surface is called the hilum. That's where everything penetrates it. Okay. Do you remember back in A&P 1, the day we started the muscular system? And do you remember there was a lot of detail to the structure of a skeletal muscle? Okay, today is like that. There's a whole lot of detail to the structure of a kidney. So it's real important to get this terminology down today, so that you can really focus later on in the chapter on how the kidney works to form urine. Okay. So if we look at various regions of the kidney, here you can see some terms: renal sinus, renal pelvis, major and minor calyces, renal papilla. I've got these things bold printed. All right, and then on this page you see several more structures. So as we look at all of these structures on this picture, okay, what you're seeing on this first picture, do you see this highlighted area there? That is that's called the renal cortex. It's just underneath the capsule. So on the picture on the right, this would be the renal capsule, and then the cortex is this underneath it. That's the cortex. And then the second picture is showing what we call the renal medulla. So it's underneath the cortex, kind of like a lot like in the adrenal gland. You have a cortex and a medulla, right? Okay, the cortex is the outer part, underneath the little capsule. All right, the medulla, though, is separated out into individual structures called renal pyramids. So each of these is a renal pyramid. The area in between the pyramids, right here, right here, right here, is referred to as a renal column. And then all of that is the renal medulla. Okay? Okay, do you see the combination of all the pyramids would be the medulla. Okay? All right, then on this view over here, you can see the ureter. Here's a ureter. And do you see how it goes through the hilum, which is the concave surface, the indention? And then do you see how it funnels? It expands out into a space. Okay. If I could remove all of that ureter and remove the blood, the renal artery and renal vein, do you see how there'd be an empty space right in here? And that empty space is called the renal sinus. Okay, so a space is, sinus is always some kind of space. Okay, so then I want you to notice the ureter itself inside the sinus. Do you see how here it's expanding? So this general area of the funnel-like structure of the ureter is called the renal pelvis. And then it separates even more. So where it separates more, like, see how it's separating up into here, and then that separates or forks. All right, this right here is what we call a major calyx. C-A-L-Y-X is singular. Okay, C-A-L-Y-C-E-S is plural, calyces. But a major calyx. And then a major calyx separates out into minor calyces. And do you see how there's a minor calyx, there's a minor calyx, there's one. And that's what goes to the bottom of each pyramid. Each minor calyx goes and forms like a cup-like surrounding at the bottom of the pyramid. Okay? So that cup-like surrounding is right there, right there, right there. And it has little holes in it or perforations. And that's what urine is going to drain through. So urine's going to form up in here, and then it drains through these little holes at the end of the minor calyx. And so that little cup-like part at the end of the minor calyx that has the holes in it is called the renal papilla. Remember, a papilla is like a projection? This is an upside down one. Okay? So that would be a renal um, papilla. I'll do it in red. Renal papilla. Renal papilla. Okay. All right. So that's just a lot of terminology that you can see. This somewhat it is macroscopic, but there's also a lot of microscopic terminology that we'll look at. Here we can see it all again. The renal capsule. That's the outer covering. This little fibrous covering. Then underneath it, all of this is the cortex, and then the medulla is all of this, made up of the pyramids and the columns. Okay. All right. And then here you can see the renal pelvis, and it separates out into major calyx. There's a major calyx, and then minor calyces. So you got that? That's not too hard, is it? You just got to sit down and learn it. All right. Now, if we could pull out, well, that's not good. Let's try this. Try this eraser again. Right. If we could pull out a section like they've shown here in this box right here and magnify it, that's what you're seeing right here. So do you see all these little yellow areas? All right, if you pulled one of those out, this is what you would see. Okay, and that is the functional unit of the kidney called a nephron. There's actually about 1 million of those in each kidney. That's what your book says, about 1 million in each kidney. So obviously this picture's not drawn to scale. They're a lot smaller than this picture leads you to believe. Okay, but do you see part of the nephron is up in, here's the cortex. And so this part of the nephron is up in the cortex, and then it loops down into the medulla. There's the medulla. And so that's within a pyramid. Okay. And the nephron is the filtering unit. Okay. And it's several nephrons empty into these big ducts right here. It may not be named yet in your notes, but I'll have it. You'll see it. But these are the ducts that drain through the openings in the renal papilla. And so they're called collecting ducts. So they collect the urine from all the nephrons and drain them into the renal pelvis through that into the minor calyx. Okay. All right. You also see there's a very extensive blood supply to the nephron, and that's very important. That's what's carrying blood a blood supply in the kidneys. And so we're going to see the blood supply associated with each individual nephron as we get on into this chapter. All right, now this picture comes from a book several editions ago, and it was replaced 2 or 3 editions ago by a picture that looks like this one. But I kind of like this older picture because it doesn't look as complicated. So you can see the nephrons a little bit better and how they're affiliated with those collecting ducts and how it drains into that minor calyx through a renal papilla. So this is a good one to look at to get your bearings on how it's all arranged. Okay. All right, and so before we get into the nitty-gritty of all of this, let's look overall at a variety of kidney functions. The kidneys do a lot more than filter the blood. That's a huge thing that they do, and that's the function that we're going to focus on in this chapter. So that's one reason I started that. Okay, but the kidneys are also involved in getting it out of the body because they're putting it in those other tubes. Okay, so that's excretion. The removal of metabolic waste from the body is excretion. Now, another function of the kidney we already studied when we did the blood chapter. Do you remember the hormone erythropoietin controls the rate of red blood cell formation in the bone marrow? And the kidneys was one of the main 2 organs that produced it. The other's the liver. Remember that? All right, so because a drop of blood runs through the kidneys about 60 times every day, isn't this a good spot to figure out if you got enough red blood cells or not? Okay, so that's where this hormone is produced. And then the kidneys produce an enzyme. I'm not going to really dwell on it as far as your test is concerned. I am going to discuss it with you as we go through the kidney chapter. It's a pretty big deal with your health, but it's called renin. And it causes a cascade of reactions to occur in your blood that ultimately affects the diameter of some of your blood vessels, which in essence controls your blood pressure. So a lot of some of the blood pressure medications affect this cascade of reactions. But anyway, we'll look at that activity um, as we go through the chapter. Then another function of the kidneys that we've mentioned in the past, just kind of briefly, is that it helps activate vitamin D that's stored in your skin, and through the action of hormones. Remember, calcitonin and parathyroid hormone, they affect this oppositely. But anyway, it helps your, in a roundabout way, kidneys help your small intestine absorb calcium from the foods you eat. And then another function, we'll look at this one in this unit, is that because the kidneys control what's in your blood directly, and because fluids don't stay in your blood all the time, they move from compartment to compartment to compartment in the body, like interstitial fluid inside your cells, the lymph, or cerebrospinal fluid. Because of that, the kidneys basically are regulating those aspects of all of your body fluids. So kidneys are hugely, hugely important. Okay. All right. So let's look at this little thing called the nephron. There you see one. And it's got two basic regions. It has a renal corpuscle and a renal tubule. The corpuscle is this rounded part. That's what receives the blood initially. And it's made up of two structures called the Bowman's capsule and the glomerulus. That's how you say that, glomerulus. The glomerulus is this little clump of capillaries right there. Okay? And so fluids are going to move from the glomerulus into this capsule that surrounds it. And that capsule is called the Bowman's capsule, or you can call it, if you want to get away from people's names, a glomerular capsule. Okay? A Bowman's capsule or glomerular capsule. And then the renal tubule has three basic regions. Two of them are curled up tubes. Those are the convoluted tubules. One is connected to the Bowman's capsule. That one's called the proximal convoluted tubule, so that would be this one. The other one is farther away. That's the distal convoluted tubule. And then in between is the loop of Henle, or the nephron loop, sometimes it's called. Okay? And that's the part that dips down into the medulla. So here's the dividing line between the cortex and the medulla. That's how it's positioned. Okay? Okay. This also, you see this picture more than one place in your notes. I forgot to remove it in one of the places, but it's okay because it's a great study picture. This also came from another book, and it just makes it all look a little less complicated than your book does. So it's a good study. It's not entirely accurate, but it works. So here you can see how blood vessels are affiliated with the nephron. Okay, and so just to review the little parts of the nephron, what's that part right there? Glomerulus. And then look at the Bowman's capsule. Do you see how it has a layer that kind of is on the surface of the glomerulus, and then it's got space, and then it's got another layer? So it's got what we call a visceral layer and a parietal layer. And if you could imagine, like if you had a balloon representing a Bowman's capsule that did not have a whole lot of air in it, but had some, and if you took your fist, the fist is going to represent a glomerulus, and pushed your fist onto the balloon and tried to move it in, do you see how part of the balloon would cover your fingers? That would be like the visceral layer of the Bowman's capsule. And then where the air is would be the space the fluid moves into. And then you have the parietal layers, the outside. So that's kind of how it's positioned. And then you see your proximal convoluted tubule, the loop of Henle, and then the distal tubule. When I said it's not entirely accurate, it has to do with, it's actually, they have found out that the loop of Henle, you've got a descending limb, and then here's the ascending limb, the fluid moves up. It actually goes all the way up before it starts convoluting. It goes up real close to the glomerulus, whereas this picture doesn't show you that. That's not a huge deal. But I also want you to notice these blood vessels. And so do you see how there's two blood vessels that connect directly to a glomerulus? Okay. One of them is bringing blood into the glomerulus, On this picture, it's the bottom one. It doesn't necessarily have to be the one at the bottom, but you see the little arrow above it. Um, it's called the afferent or afferent arteriole. That carries blood to something. Afferent means toward. Efferent means away from. So the afferent arteriole is bringing blood to the glomerulus. And then the efferent arteriole is this one, and it's bringing blood away from the glomerulus, and that connects to a bunch of capillaries that surround the nephron tubule. Okay? And they're called, it's real hard, peritubular capillaries. Okay? In your book, you will also see that an extension of the peritubular capillaries surrounds the loop of Henle. For us, we're going to still call that peritubular because they are, but sometimes they can be further recognized as the vasa recta. That's not in your notes, but it is in your book. Okay, you'll see that. All right, and what's going to happen? I'll just go on and kind of tell you. When urine is formed, it's going to be formed through 3 stages. The first stage is when substances are moving from the blood into the glomerular capsule, and we'll look at that process today. And then that fluid that moves is not called blood plasma anymore. See, the cells don't move, so blood cells don't get across. Large molecules like large proteins don't move, but everything else can. So basically, this filtrate that is now in the Bowman's capsule is the same composition as interstitial fluid or lymph, because it's everything in the blood plasma except the big proteins, pretty much. Protein content's different. But that filtrate now is going to start moving through the proximal convolute tubule, loop of Henle, distal tubule, until it gets to, here's a collecting duct right here. But did you catch that the composition of this filtrate at the Bowman's capsule is like interstitial fluid, which is like blood plasma except for the protein content? So that means it's got a lot of things in it you still need, like glucose, maybe some calcium, maybe some sodium. All right, so as that filtrate now moves through the rest of the nephron, the things that are in it that you still need to keep are going to move into the peritubular capillaries. Sometimes something that you need to get rid of doesn't filter initially. And so do you see how that becomes part of the blood in the peritubular capillaries? So those substances will move from the peritubular capillaries into the tubule. So by the time it makes its way to a collecting duct, it's all filtered up. Okay? And it's only got in it the things you don't need anymore. Okay? Except for CO2, but in the meantime, any of the kidney cells' CO2 have moved right there into this blood of the peritubular capillaries. Okay? One thing that is super interesting to me about this blood supply is if the glomerulus is capillaries, which it is, have you noticed that it's not like other capillary beds we've talked about? It doesn't have an arteriole on one end and a venule on the other end. It's got an arteriole on each end. If you notice the picture of it at the glomerulus, the blood's not changing color, is it? Meaning there's no gas exchange there. So there's an arteriole on each end, and then the efferent arteriole leads to peritubular capillaries, which do involve a gas exchange with the kidney cells. Okay? And then they're going to lead to venules and veins that are going to lead out of the kidney. That's interesting to me. Okay. So here you see hanging down from the stomach, that's the greater omentum. You've already been tested on that. If you remove that, you see the intestines. And if you remove all of that, along with the parietal peritoneum, there you see the kidneys and the blood vessels associated with those. Okay. All right, and then here you see it sectioned. And so, on this actual kidney, if you look real close, you can actually see the pyramids. You can see major and minor calyces, cortex. All right, as you can see there. So those are good to study. You may not have those pictures in your notes. They're coming from a cadaver site that we can utilize. But those are good. I know you can't see it well in your notes, but you can go home and look on your computer screen and see that large if you want to. And there you see it again. I have got, your notes are just loaded with pictures of the kidney. This is very important. There are a lot of questions on the test that involve structures associated with the kidney. Okay. And so you can review. Okay. All right, now we're going to look at the blood supply to the kidney. And you thought you already looked at it, but now we're going to look at all the blood vessels up until the afferent arteriole. Okay. So the kidneys are fed by the renal artery coming off the abdominal aorta. And then here I've got a list, and I have this list in more than one place, of the flow of the blood to get from the renal artery to the afferent arteriole. And then we're going to go through the glomerulus and the efferent arterioles, peritubular capillaries, and then a bunch of veins to get to the renal vein. Okay, and so you can see that flow, and if I'll try to show it in green on this picture. It's hard to see some of it on the picture because there's so much redness to the picture anyway. So it's hard to pick out the arteries because they're red. But anyway, here's a renal artery. And where it branches, and it doesn't show the completion of the branches, but as it branches into a column, where I'm drawing green is on top of a little red line in that picture. And that is called an interlobar artery. Interlobar artery. And then from there, it branches, so it goes on top of a pyramid. So like right here, right there, right there, right there. And that's called the arcuate, or it's also referred to as the archiform artery. So that's what's on top of a pyramid. And then if you'll notice, branches from it go out into the cortex, like here, here, here. Those are called the interlobular, not interlobar, but interlobular. And then they've renamed those, of course, they rename all this stuff. Interlobular, or, this is more descriptive, cortical radiate. Radiates out into the cortex. Cortical radiate artery. So that would be these little ones here. Okay. And the afferent arterioles come off of those. Okay. So in this picture on the right, or in the middle here, the picture in the middle, here you're seeing a cortex and a medulla. Okay, so imagine a blood vessel coming up just outside the pyramid. That would be the interlobar. And watch. This one, that artery right there, is the arcuate or archiform. And then this one is the interlobular. Isn't a lobule smaller than a lobe? Okay. The lobular artery is smaller than the lobar, okay, and more numerous. So here's the interlobular or cortical radiate, and then the afferent arterioles come off of that, okay, to go to a glomerulus. Okay. So here you see another view of that, and it's all labeled for you. If you look carefully, you can see these labels. I'll try to highlight them in green again. What is this one coming up in the column? Interlobar. And what is this one? Arcuate or archiform. It's the arch, you know. All right, and then so the one, I can't see it on here, coming out this way. Cortical radiate, doesn't show it too well here. And then the one coming off of that, you can't see it close up, would be the afferent arteriole. I'll have a closer picture of that. Here's that old picture that I love so much. Okay. All right, if I had a blood vessel coming up here outside the pyramid, that is interlobar. And then this one, archiform, interlobular. So this is the cortical radiate right there. Okay, now, here you can see the afferent arteriole coming off of it, going to the glomerulus, efferent arteriole coming out of the glomerulus, and then that leads to peritubular capillaries, all of these, and then they pull together into, now this is easy, here's the veins, interlobular vein, archiform vein, interlobar vein that goes to the renal vein. Okay, so at least the veins have the same names as the arteries that they parallel. Okay, but you need to know that flow. And if you didn't get it, there it is again. Okay, I've got it in a lot of places. It's important to know that flow. In your lab, you'll be going through this again. Okay. So here on this picture, this is showing you that term. I probably should test you on it, but the vasa recta, that's just an extension of all the peritubular capillaries around the loop of Henle, but we're going to still just call them peritubular capillaries. Okay. All right. This is the one I meant to remove. Sorry, it's in your notes. I like it. This is a neat picture because this is showing you more details at the renal corpuscle. Remember, the corpuscle is the glomerulus and the Bowman's capsule together. And you see the visceral layer of the Bowman's capsule. Do you see how the cells have these little extensions and there's spaces in between them? All right, and that's one of the reasons it's very porous here. And that's one of the reasons substances can easily move across that layer into the space of the Bowman's capsule. Another reason is that the glomerular capillaries themselves, you don't see it on this picture, but they have extra pores in them. So it's very, very porous. It's very easy for substances to move across. They're not so porous that cells or platelets or big proteins move across, but everything else can very easily. And then on the right, you're seeing an electron micrograph, a scanning, color-enhanced electron micrograph that is showing those cells. You don't have to name the cells. They're named there, the podocytes. Don't worry about that name, but it gives you the idea. That's that visceral layer. It's kind of strange looking. But, okay. So if you haven't gotten the structure of the nephron, there it is. And this is a more accurate view of a nephron because it's showing the loop of Henle's ascending limb going way up high, close to that afferent and efferent arteriole of that nephron. Whereas that picture I put in your notes so many times doesn't show that as well. It shows the distal tubule being there. But notice on this picture the afferent and efferent arterioles. Do you notice that the afferent arteriole is bigger in diameter than the efferent? Not a whole lot bigger, but a little bit. And on a lot of your pictures, you'll see that. That is extremely significant, that the afferent arteriole is bigger than the efferent. And we'll see why when we look at urine formation, but it's bigger. So if you didn't see arrows to know where the blood was flowing, what direction it was flowing, if you just looked at the diameter of those two blood vessels, the bigger one is the afferent arteriole. And it says that in your notes as you're reading through. Okay. Okay. All right. And then this is a very modified view of the nephron. So here you see blood flowing into the nephron by way of the afferent arteriole. And then it leaves the nephron, blood does, by way of the efferent arteriole that leads to the peritubular capillaries. And then what comes out of the blood at the glomerulus goes through the renal tubule. Okay, and then we're going to see that there's some processing as that filtrate goes through the tubule between the filtrate and the blood of the peritubular capillaries. And then when it's all said and done, it ends up at a collecting duct. Okay. This is a micrograph, which you'll see kidney slides in lab. And the cortex and the medulla look very different under the microscope. You can actually see the renal corpuscles in the cortex. So I see a renal corpuscle there, I see one there, I see one there. So that's the glomerulus and the Bowman's capsule. You don't see that on the picture on the right. That's showing the medulla. All you see is a bunch of tubes. So, okay. All right, now this is a further flow chart and as everything moves, it starts off in the blood. This is for urine, where urine comes from. It comes from the blood of the glomerulus, and then from there substances move out of the blood to go into the Bowman's capsule, and then from there, proximal convoluted tubule, the descending, then ascending limbs of the loop of Henle, and then distal tubule, collecting duct, and then from there, it's flowing away from the kidneys, into the minor, the major calyx, into the renal pelvis, ureter, bladder, urethra. So that's the flow of the urine and as it's formed. Okay, now here's a weird structure associated with the kidney. It's called a juxtaglomerular apparatus. The prefix juxta means next to something. Okay, it just means next to. So this is something next to the glomerulus. Okay, we're going to look at what it's made up of right now, and then as we look at urine formation, we'll see its function. Okay, but it's where the top of the ascending limb of the loop of Henle comes up between the blood vessels connected to the glomerulus, comes up between the afferent and efferent arteriole. And it basically is going to, in a roundabout way, it controls the diameters of the afferent and efferent arteriole, which in turn affects how much blood's going into the glomerulus at each moment. Okay, there's two groups of cells at the juxtaglomerular apparatus. One group is called the macula densa. One group is called the juxtaglomerular, which means next to the glomerulus. Juxtaglomerular cells. So here you can see it. The juxtaglomerular apparatus, if it's in this area, so here you see it magnified. This is the very top of the loop of Henle, and there's some real large, tightly packed cells right there. Those are the macula densa cells. And then the afferent arteriole has some special smooth muscle cells in its wall. Those are the juxtaglomerular cells. Okay, so those two groups of cells, the macula densa and the juxtaglomerular cells, together make up this structure called the juxtaglomerular apparatus. Okay, and later this morning we'll see how it affects the flow of the blood. Okay, all right. So we've looked a whole lot at the structure of a nephron, but there's basically two kinds of nephrons: a cortical nephron, and then here's that prefix again, juxtamedullary nephron, which has nothing to do with the juxtaglomerular apparatus. It just has the same prefix that means next to. Okay, so obviously, evidently, I guess the same person named both of those structures. They like that prefix. But I don't think it's in any other chapter of this book, but it's associated with kidneys. All right, anyway, the cortical nephrons are ones that have a fairly short loop of Henle, and they sit up high in the cortex. So this one is a cortical nephron, whereas this one that has a longer loop of Henle and that is closer down to the medulla, that's a juxtamedullary nephron. The longer a loop of Henle is, the more water can be reabsorbed from the filtrate and put back into your blood. And so those juxtamedullary nephrons are really associated with water reabsorption. It's interesting to me that mammals that live in desert environments have a much higher percentage of juxtamedullary nephrons than mammals that don't live in desert environments. They can retain water better. Okay. Mammals that live in desert environments, the longer the loop of Henle is, the more water can be reabsorbed from the filtrate. Okay. You're welcome. All right, now we're going to look at what happens. I've told you some of what happens. You basically know the flow. But when urine is formed, I've mentioned to you the fluid from the blood moves into the nephron itself, into that Bowman's capsule, and now it has a new name. It's called filtrate. Okay, and then once it gets through the nephron, and it's in the rest of the organs, it's referred to as urine. After it gets processed, it's referred to as urine. The 3 steps involved in this urine formation you can see listed here. I have mentioned those to you without using the names, but we're going to look at these three steps. Um, on the top picture, you can see regular capillaries in the systemic circuit where you have an arteriole at one end and a venule at the other, and gas exchange along with other exchange occurred there. Okay. But at the kidney, you've got two types of capillary beds. You've got the glomerular capillary bed, and then you've got the peritubular bed. Okay. So watch this. Here's the three steps of urine formation, just listing, not details of them. As blood comes in to the afferent arteriole, it goes into the glomerulus. Substances move out of it. Pretty much anything that's in blood plasma can move. Not everything moves, because then you'd have nothing but cells and big proteins left over and it can't go anywhere. But this is constantly flowing from the afferent into the efferent. But as it does, a lot of the substances from the blood plasma move into the Bowman's capsule. That's called filtrate. And that's called glomerular filtration, that process. Okay. And so the composition, this is significant, the composition of this filtrate initially is pretty much the same as blood plasma except for the protein content. So it still has in it a lot of things you still need, like water, a lot of water, maybe some sodium, a lot of glucose, a lot of different things you still need. And so as it then moves through the tubules, the things that you still need are reabsorbed into the peritubular capillaries. That's step 2, tubular reabsorption. Okay? But if you go back to the glomerular filtration, not everything is going to get filtered that you still need to get rid of, because some of it remains in the blood at the efferent arteriole. Okay, and so if you still need to get rid of it, now it's going to be secreted into the tubules. Okay, and that's the third step, tubular secretion. I like to make an analogy of urine formation with, sounds so silly, but it makes sense to me. There used to be a show on HGTV called Clean Sweep. I don't know if any of you ever saw that. But in that show, they would go into someone's home, and they just had so much clutter. It was unbelievable. And they were going to fix that. And the way they did it was initially they did 3 steps. Just like urine formation is 3 steps. The first thing they did was, let's say if it was the living room they were going to fix, they got everything they could find out of the living room and they put it outside under a tent. Even stuff that family still needed, but they needed to get it all out and then sort through it and put back in the things that they still needed. So as they pulled everything out of the living room, that would be like glomerular filtration, getting things in a place that then you can pick and choose what you want. Okay, that's filtration. All right, and then putting the things that you still want back into the house, that would be like reabsorption, tubular reabsorption. But usually when they pulled things out initially, they left something behind like in a closet. And so as they were putting things back in, they noticed, oh, I forgot about that I don't want anymore. And so they pulled it out. And so that would be like tubular secretion, the things that remained in the blood that you need to get rid of. Okay. And so those are the 3 steps. So that makes sense to me. I loved it. When that show came out, the kidneys made sense to me. So anyway. All right, here's a better view of this is not a word, porousness of glomerular capillaries. See all the extra little pores in it? They're called fenestrae. You don't have to name that for your test. But it's very, very porous. So that means filtration is rampant there. There's a lot of filtration. Okay. So now we're going to look at the details of glomerular filtration. And the big deal is the afferent arteriole is a bigger diameter than the efferent. Listen to me. If the afferent is bigger than the efferent, does it make sense to you that the hydrostatic pressure in the glomerular blood is higher than hydrostatic pressure in any other blood capillaries of the body? Because the afferent's bigger than the efferent. That bumps up the blood pressure of the glomerulus. Okay? And that pressure that extreme blood pressure of the glomerulus is the driving force for filtration. It's just not normal little everyday filtration. It's whoop, massive filtration can occur because the glomerular hydrostatic pressure is high. And the reason it's high is because the afferent arteriole is bigger than diameter than the efferent. Okay? And so, and because also the glomerular capillaries are permeable, a ton of that stuff moves across. And once it does, and it's in a Bowman's capsule, it's referred to as filtrate. What's the composition of filtrate? It's like blood plasma. Like if it's 10% glucose in the blood, it's going to be 10% glucose in that initial filtrate. It's just like blood plasma except for the presence of the big proteins. Okay? Little proteins might go across, but for the most part, the proteins don't go across. Okay? All right. So there's no cells in it. That's not blood plasma anyway, cells, right? So there's no cells. There's no red cells, white cells, or platelets. All right, but there are 3 pressures that affect filtration. And here's where your study in the past of hydrostatic and osmotic pressure gets real, is at the kidney. Okay? All right, there are 3 pressures involved that determine the overall pressure called the filtration pressure, which is going to regulate how fast the filtration occurs. I've initialed the pressures. Here you can see them on this page. You have glomerular hydrostatic pressure, GHP. So that's the pressure due to the presence of the water or the blood pressure in the glomerulus. The higher it is, the more filtration will occur. Does that make sense? That's because it's a pushing pressure. All right, and then there's glomerular osmotic pressure. That's due to the presence of all the things dissolved in the blood plasma at the glomerulus. The higher that is, the less filtration will occur. Remember, an osmotic pressure is a pulling pressure or keeping pressure for water? Okay. And then there's also fluid pressure in the Bowman's capsule, and that's called capsular hydrostatic pressure. The higher it is, the more that would decrease filtration because that would push it back into the glomerulus. Does that make sense? That normally doesn't get high because that fluid is going somewhere. It goes into the proximal convoluted tubule to relieve it. You know. But so basically of these 3 pressures, only one of these favors filtration, and that's this one. The glomerular hydrostatic pressure favors filtration. The other 2 oppose it. Does that make sense? You can see that on this picture. I like this view. Okay. Here, do you see this big yellow arrow? That's representing glomerular hydrostatic pressure. And then the orange arrow is representing glomerular osmotic. Here it says plasma colloid osmotic. That's a fancy name for glomerular osmotic pressure. Do you see it? Because the arrow is moving in the opposite direction, that means it opposes filtration. But because it is less, it's a smaller arrow than the glomerular hydrostatic pressure arrow. So if you just had those 2 pressures, filtration would occur, right? Okay. But you have a third pressure, and it opposes filtration. It's that one. It's the pressure due to the presence of the fluid in the capsule. All right. If you added up this one and this one, the sum of them would be less than this one. And that means filtration is going to take place. And you can see that in the numbers on the bottom. So if it says the glomerular hydrostatic pressure is 60 millimeters of mercury, that should be mercury, millimeters of mercury. Millimeters is a unit of length, isn't it? All right, it should be 60 millimeters of mercury. And then the reason the other 2 pressures have a negative sign in front of them is meaning that they oppose the glomerular hydrostatic. But even like the little math equation I put on a couple of slides before this, I said the filtration pressure is equal to the glomerular hydrostatic pressure minus the sum of the other 2, glomerular osmotic plus capsular hydrostatic. Okay, so if we put these numbers in there and forgot the minus signs and plus signs, we just made glomerular hydrostatic 60 glomerular osmotic 32, and glomerular or capsular hydrostatic 18. If you add up 32 and 18, that's 50, and 60 minus 50 is a plus 10. That means our filtration pressure is plus 10. It's positive, and that means filtration takes place. Okay? Do you see how any body condition which would affect these three pressures is going to affect the rate of filtration? Okay. So think about this. If you ever heard that really high blood pressure can affect the kidneys? Because it's going to up that glomerular hydrostatic pressure so much that it literally could blow out the glomerular capillaries, which could cause kidney failure. Have you ever heard that in a severe drop in blood pressure can cause kidney shutdown? It's because it drops the glomerular hydrostatic pressure so much that it can't override the other two, and it backfires. Do you see? Filtrate would move into the blood at the glomerulus. Okay. Thank goodness we have an autoregulatory mechanism that actually keeps the filtration pressure pretty constant, even though our body experiences changes in blood pressure. And that is the function of the juxtaglomerular apparatus. Okay, I have just enough time to go over this. That's good. Okay, so the juxtaglomerular apparatus is the structure that actually regulates, it basically regulates glomerular hydrostatic pressure to keep your filtration rate right at plus 10, so that filtration rates are pretty constant, even though we have fluctuations in our blood pressure. All right, and the way it does that is through the macula densa cells. Okay, the macula densa cells, they are cells at the top end of the loop of Henle, the ascending limb of the loop of Henle, where it comes up and touches the wall of the afferent arteriole. Okay, the macula densa cells, here they are, they recognize the concentration of ions in this filtrate that's in here. So remember, this filtrate has come from here, and it's gone through the proximal loop of Henle, and then loop of Henle, and so there it is at the top of the loop of Henle. Let's say filtration rate for one reason or another has slowed down. Do you see how ion concentrations then will be less at this point? And the example I give you in your book is chloride ions, but it's other ions too. So if the macula densa cells recognize a lesser amount of ions, then that's going to signal the smooth muscle cells, these juxtaglomerular cells of the afferent arteriole, to relax. If they relax, what happens to the diameter of the afferent arteriole? It gets bigger. So that means more blood's going to flow in, which is going to do what to the glomerular hydrostatic pressure? It's going to increase it, right? And then as these juxtaglomerular cells relax, they release into your bloodstream this enzyme called renin. And renin sets off a cascade of reactions in blood vessels or in your blood. I'm not going to test you on the cascade, but I want to talk about it here. Okay. So renin, when it's released by the juxtaglomerular cells into your bloodstream, it activates a protein that's called angiotensinogen, and it changes it into one called angiotensin 1. And then there's another enzyme coming from your lungs called angiotensin converting enzyme that converts angiotensin 1 into angiotensin 2. And here's the bottom line. You have receptors for angiotensin 2 in a variety of blood vessels in the body. And when angiotensin 2 combines with those receptors, that blood vessel constricts, which ups your blood pressure. But one of the blood vessels that responds to angiotensin 2 is the efferent arteriole. So while the afferent arteriole is relaxing, the efferent is constricting, which further ups the glomerular hydrostatic pressure to keep that filtration rate going. Okay? Angiotensin 2 does other things too, and it tells you all that here. You don't have to know that for the test. But one of the blood vessels that constricts is the efferent arteriole. So in your notes, it tells you that renin basically causes the efferent arteriole to constrict. So afferent dilates, efferent constricts, which ups your glomerular hydrostatic pressure. Some hypertension medications are called ACE inhibitors. Some of you have heard of that. ACE means angiotensin converting enzyme. So it inhibits that, which prevents this, which means it lowers your blood pressure in your body, because there's other blood vessels involved in that constriction. Okay, and that's how an ACE inhibitor works. Okay, this is my favorite table in your entire book. I love it. On the left, the 180 liters, that's how much filtrate is produced every day on the average in a person. That means how much stuff is filtering into Bowman's capsules. Can y'all visualize 180 liters? That's like 50 gallons or 92-liter Coke bottles. Do you urinate that much? No. This is how much you urinate every day. So even though we've talked a lot about filtration, urine formation is not anywhere near finished. We got to reabsorb. Reabsorption is involved in getting it from there to there. Reabsorb a whole lot of things, especially water. Okay? If you didn't have all the reabsorption, you'd dehydrate just like that in just a moment. Okay? All right, so you've got a lot to study, to chew on these next few days. When we get back next time, we'll finish urine formation, and then we'll go through the other organs of the urinary system. And then we'll do the next chapter.